Hi, and welcome to another installment of an R&R production. Today we're covering meiosis. This is a cell interphase. From here you see that the DNA has been replicated from 46 to 92 chromosomes, enough for meiosis. Now you see the nuclear membrane start breaking down and also the chromosomes starting to coil. You also see the centrioles appearing. Now you see distinct chromosomes and also spindles starting to form all the centrioles. Another phase of prophase 1 is the crossing over between two chromosomes. Here you see the legs of chromosomes crossing over. This is for genetic exchange of DNA. Now we are in metaphase 1. We see the chromosomes lining up across each other and the spindle fibers extending to allow them to line up along the equator of the cell. After metaphase 1, you see anaphase 1. You see the crossed over homologous chromosomes being pulled apart via the centrioles and the spindle fibers. Here, a continuation of anaphase 1, you see the homologous chromosomes being continued to be pulled apart. Here you see anaphase 1 has concluded and that telophase 1 has started. Here, a continuation of telophase 1, you see the nuclear membrane starting to reform and the chromosomes starting to uncoil. Continuing in telophase 1, we see a cleavage furrow start to appear, and also the nuclear membrane has totally formed along with the DNA totally uncondensing. Here, we see two distinct cells have formed, and telophase 1 has ended with the process of cytokinesis.
Now welcome to Meiosis 2 introduction. Please note that Meiosis 2 occurs in two cells, but we are only showing one. Here's a cell following Meiosis 1. It has 46 chromosomes with 2 chromatids. Here we have a cell undergoing prophase 2. The nuclear membrane is breaking down, the centrioles are moving to the poles, and the chromosomes are condensing. Continuing prophase 2, you can see the chromosomes have completely coiled and the centrioles have begun to form spindle fibers. Now you see metaphase 2. The centriole fibers have completely formed and the chromosomes have moved to the equator. Now during anaphase 2, the chromatids split as the spindle fibers pull them towards the opposite poles. Now continuing anaphase 2, the chromatids have completely been pulled to the pole as the spindle fibers have shortened. Here you can see telophase 2 beginning as the cleavage burrow begins to form and the spindle fibers begin to break away. Continuing with telophase 2, you can see the nuclear membrane begin to form, the chromosomes begin to uncondense, and the cleavage burrow form even more. Continuing telophase 2, the nuclear membranes have fully formed, the chromosomes have fully unwound, and the cleavage burrow has continued to form. Here you can see telophase 2 is completed as cytokinesis has split the two daughter cells, which are identical.
After meiosis 2 is concluded, we have 4 identical daughter cells from the process of meiosis. From here, each cell has 23 chromosomes and can either become 4 sperm in a male or 3 polar bodies in an egg in a female.